Yes, good evening to all of you. My voice is audible. Hope my voice is audible to all of you. My voice is audible. Please let me know. Hello. So my voice is audible. So yes, today we have to discuss further. Yesterday we have discussed nucleotides, nucleosides, DNA structure and RNA. Yes. Hello Shivangi, how are you? And even we have done mitosis also. So we have covered, I think we have just covered yesterday DNA and RNA structure, right? So we have covered DNA and RNA structure. So I am correct? Yes, good evening Pooja. So we have just covered DNA and RNA structure. So I think we have just completed this PPT yesterday. Yes. So we have covered only RNA and DNA structure yesterday. And maybe today we have to start with DNA replication, transcription and translation, right? So we have to cover replication, transcription and translation today. Yes, good evening Bhavni, good evening all of you, Shivangi, Pooja. Just let me know, we have to complete today transcription reading. We already covered this part also. One more thing is there which is in your syllabus, which is gene structure. So if you are able to see here, find structure of a gene. So I will try to complete this gene structure today. Okay, I will try to complete this fine gene structure today. Yeah, this one. I think I also. Then we have a regulation of gene expression also. Yes, good evening to all of you. How are you all? So have you done your yesterday's homework, all of you? Yeah, good evening to all. So have you done your yesterday's homework? We have to discuss protein, not protein, sorry, we have discussed DNA RNA structure. So have you completed it or not? Please let me know. No, no, I will reply to all of you, but I will read your mail. Then I will definitely mail you back also. I will reply on the mail also. Okay. So today we have to start another lecture of building the on fine structure of a gene. So I'm just waiting for 1 or 2 minutes more so that students can join. In the meanwhile, if you have any questions, please ask. If you have any query or questions, then please ask. Otherwise, I will start the session. Okay. So I'm audible to all of you. Please let me know. Please let me know whether I am audible to all of you or not. Yes, Tanisha, try to do it on a same day because then it becomes very difficult for me to maintain the record. So I request all of you to submit the assignments on time. You have to submit it only for up to the 15th of December, then you will be free. So try to submit it on a time as soon as possible. So my voice is audible to all of you. Please let me know if you have any query or something so that we will start the class. Please reply all of you. I request all of you to reply. Just waiting for 1 or 2 minutes more, otherwise I will start the session. Those who will left today, they will watch it later also, no worry. So I am just going to start the session. Now you are getting my voice? Hello, my voice is audible to all of you. Please let me know. Are you able to get me properly? All of you, are you able to get me properly? Please let me know, Bhavni, so that we should start now. So shall we start now? Okay. So I request all of you to keep your earphones along with you. If you will not be able to get me, then you can. Okay, okay. Okay, so thank you all. So we will start today the topic which is here, which is fine structure of a gene. I'm taking this topic because this is very simple topic and this is associated with this lack of brain and regulation of gene expression. So I will start with this topic. We already completed these topics. Only transcription and translation is left. Transcription and translation, replication, transcription and translation is left. So I start with the gene. Then we will move from 8, 7, 6. We will try to these together. Okay. Then after it we will do the linkage also. So I think I have to start now. I will try to wrap up your syllabus by this weekend, and I will try to take little bit long classes also. So now I am just going with the gene structure. Okay, so I am just sharing the gene structure. Yes. So yes, today we have to study the fine structure of a gene. Basically, what is the gene? We have to understand the history of a gene. Then we have to know we have two types of genes, basically prokaryotic as well as eukaryotic. We must know the structure, the features which are there of a particular gene. So basically, let us start now. So a gene is a specific sequence of a DNA containing the genetic information required to make a specific protein. So gene is a specific sequence. Gene is a specific sequence of a DNA containing genetic information required to make a specific protein. So prokaryotic gene is uninterrupted. So we have two types of genes. So prokaryotic genes are uninterrupted. In eukaryotic genes, the coding sequence are separated by non-coding sequences and they are known as introns. Just give me one. So, in eukaryotic genes, the coding sequence which is known as axons, they are separated by the non-coding sequences which are known as introns. So, in case of eukaryotic organisms, we have axons as well as introns. So, in complex eukaryotes, introns account for more than 10 times as much DNA as axons. So this is the history of genes. The classical principle of genetics was disused by Mendel in 1865 on the basis of breeding experiments with peas. He assumed that each trait is determined by a pair of inherited factors, or you can say characters, which are now called a gene. So these pair of characters are now known as genes. So Mendel's work was, as I already told you, he was not appreciated at that time. So Mendel's work was rediscovered in 1900 by Hugo de Varis, Correns and Teschmark. So these were the scientists who rediscovered the work of Mendel. Then again in 1909 there is a scientist William Johnson who coined the term gene. So gene is there from Mendel, but we are not able to know at that time. At that time it is known as characters basically, but it is basically the genes who control the particular character or a trait. Then in 1905 we have William Weissman who coined the term genetics. In 1972, Walter Weers and his team determined the sequence of a gene in bacteriophage MS2 cause protein. According to Roberts and Sharp, this is Roberts and Sharp, the gene can be split into segments, making it possibly that a single gene might be coding for several proteins. So they were the one who tell us about the split genes in 1978. Now what is gene basically? What is a gene? Any one of you? What is basically a gene? Please let me know what is gene basically. What is the gene? Let me know about genes. Yes, Pooja is structural unit of heredity. Sequence of DNA code for a particular character. Okay, very good, very good. So genes are a unit of heredity. Very good, Sheikh. So all of you please reply what is gene. All of you must reply. So we are just dealing with the gene. So what is basically a gene? The gene is a functional unit of heredity. So this is a cell. Inside the cell we have a nucleus. In the nucleus we have a chromosome, and on the chromosomes we have the double helical structure which is known as a DNA, which will by translation give us the proteins. So this is particularly the segment of a DNA. So each gene is a segment of a DNA that give rise to a protein product or RNA. A gene may exist in alternative forms known as alleles. So Mendel was also able to tell us about alleles. So a gene may exist in alternative forms which are known as alleles. The chromosome in fact carry the genes. So the genes are present on the chromosomes basically. So each chromosome consists of linear array of genes. So genes are present on a chromosome. Now we have two types of gene structure. We have prokaryotic gene structure as well as we have the eukaryotic gene structure. Now what is there in the case of prokaryotic genes? So the DNA genes, based on their activity, we have two types of genes. We have housekeeping genes as well as we have specific genes. Now what are the structural feature of this housekeeping or specific genes? These are the simple gene structures, small genome. Prokaryotic genes are collinear with the protein. So we have the coding region as well as we have the promoter element, terminal region or the terminator. So we have the coding region, the starting point basically where there will be the initiator codon, and we have the terminator region also. So no introns, there is no introns in between in case of prokaryotic gene structure. You have to keep this thing in mind that in case of prokaryotes there is no introns. So we have the starting region, we have the terminating region, and in between we have the coding sequences which will code the protein that is we need. Or you can say from the DNA we will get the RNA, and from this RNA we will get the polypeptide. So from this DNA we are getting RNA, and RNA too we are getting the polypeptide. So this is the coding region. Okay. So we have the coding region, promoter elements and terminal region. Now what is promoter elements? The upstream elements from the start of the coding region includes the promoter sequences 50 to 100 upstream of the start codon. We have the initiation site or the start site. So we have the initiation site just before the coding sequence. So before the coding sequence we have the initiation site. Now any nucleotide present on the left is denoted by minus symbol, and the region is called upstream element. So we have one upstream element also, which will be there before the coding sequence, which will be by minus sign. So any nucleotide present on the left is denoted by the minus symbol, and the region is the upstream element. Start symbolized by +1. Any sequence right to the start is known as downstream element and now termed as positive. Now let me show you. So some sequences are there before the coding region. These sequences are there in the minus, and these sequences which is present at -10, this sequence is known as TAT or Prebino box. So what is this Prebino box? I will tell you. And at the -35 region we have another sequence which is TTGACA. So these two are the most important promoter elements. At -10 region as well as at -35 region we have the promoter elements. They are recognized by the transcription factors. So we have the transcription factors also, which will be recognized only by these sequences, only by these promoter elements. At -65 to -60 we have activator elements. These are promoter elements. Then we will have at -60 to 65 we have the activator elements. At -200 to -1000 right here we have the enhancer sequences. So we have promoter sequences, we have activator sequences, as well as we have enhancer sequences, but right before the starting sequence, right before the initiation sequence, right before the coding sequence. All these are present just before the coding sequence. Getting my point? Then after it we have the last one, which is the terminal region. We have the terminal region. So you have to keep it in mind. We have the coding region, promoter region and terminal region. Now what will be there in the terminal region? Sequences for the termination of transcription. It takes place by root dependent mode or root independent mode. So terminal is basically at the end of the gene. So this prokaryotic gene structure, are you able to get it? This prokaryotic gene structure. After it we have eukaryotic gene structure. So this is only the prokaryotic gene structure in which we have coding region, promoter region and terminal region. After it we have the eukaryotic gene structure which will be little bit more complex as compared to prokaryote. Now what will be there in the eukaryotic gene structure? In it we have exons, introns, promoter sequences, terminator sequences, upstream sequences, downstream sequences, enhancers, silencers and signals. So let us start now. So what are basically exons? So exons are the coding sequence transcribed and translated coding for amino acids. So these are the coding sequence. Like in case of prokaryotes, introns are absent. Only coding sequences are present. But here we have exons as well as introns. So coding sequence transcribed and translated coding for amino acid in the polypeptide chain vary in length in number, sequence and length. A gene starts and ends with exons. So what are basically introns? So coding sequences are separated by non-coding sequences. So here what is basically happening? We have a coding sequence which will code for the protein, and we have some non-coding sequences also. So any nucleotide sequence that are removed when the primary transcript is processed to give the mature RNA are called introns. So all introns share the base sequence GT in the 5' end and AG in the 3' end. So these introns, which are non-coding sequence, they were discovered by Sharp and Roberts in 1977. So if you are able to see, these red one is the coding sequence, this blue one is the non-coding sequence. So we have to cut these non-coding sequence first, so that we will get only exons, or we will get only the protein coding sequence. So this is the coding sequence, this is the non-coding sequence, and we have 5' end and we have a 3' end. So if we are just allowing the promoter to start the reaction, then what we will see by after transcription, what is basically transcription? From the DNA we will get the mRNA. So what will be there? We will have a poly A tail at the terminator side. Please keep this thing in mind, that on the terminator side we have a poly A tail, and in the promoter side we have a 5' cap. Okay. So now what we have to do? We have to remove these introns. So when these introns will be removed, only exons are there. It is known as the protein coding sequence, which will later lead us to the polypeptide. So from the DNA we will get the pre-mRNA, from it we will get mRNA and polypeptide. So now what are promoters here? Promoter is a regulatory region of a DNA located upstream controlling the gene expression. So promoters are always just before the reaction, before the coding sequence. It will always be at the 5' end. So core promoters will have the start site at -34, binding site for RNA polymerase. So general transcription factor bind site. So proximal promoter contain primary regulator element approximately 250 specific transcription factor binding sites. One more thing which we have there in the eukaryotic gene, we have a Tata box. Like in this case we have the Prebino box. Similarly, in case of eukaryotes, we have the Tata box, also known as Hogness box, which will be there at -30 to -80 side, and these are the sequences which help in the transcription and translation. So these together are responsible for binding of RNA polymerase 2, which is responsible for transcription. So upstream we have 5' end. So 5' end has several functions including mRNA transport and initiation of translation. So you have to keep one thing in mind, that at the 5' end we have the cap. So at the 5' end here we have the cap, which is 7-methyl guanine. So this is 7-methyl guanine. And at the end we have the poly A tail. So at the 5 prime end we have cap, and this cap facilitates the initiation of translation. So when we will do the translation, then you will keep all these things in mind, that the cap facilitates the initiation of translation, and in the downstream we have the poly A tail. So the translation termination codon TAA, this sequence signal for the addition of poly A tail. So if you will able to see here, this is the upstream region, this is the downstream region, this is the 5 prime end, this is the 3 prime end. So at the 3 prime end we have a poly A region, at the 5 prime end we have the cap, 7-methyl guanine cap. Even we have the promoter boxes also, we have Tata box, we have cat box. All these promoter region, they will initiate the reaction. So we have axon, and in between the axons we have introns. But we have only to take axons only. So we have to remove these yellow one non-coding sequences before. Getting my point? Then at the end we have a terminator which is recognized by the RNA polymerase as a signal to stop transcription. Enhancer is there which enhance the transcription of a gene. Silencers are there which will reduce or shut the expression of a nearby gene. So all these are the components of the eukaryotic gene structure as well as the prokaryotic gene structure. So this is your homework also. You have to draw eukaryotic gene structure as well as prokaryotic gene structure. You have to draw only, not to write. You have to draw only. Now why there is a need of introns in between? Why there are introns in between when they have to be removed out? So introns do not specify the synthesis of proteins but have other important cellular activities. So many introns encode RNAs that are major regulator of gene expression. It contains regulatory sequences that control transcription and mRNA processing. So introns allow axons to be joined in different combinations, resulting in the synthesis of different proteins from the same gene. So they are helpful in transcription. They are major regulator of gene expression. They do not specify the protein, but they have some another functions also in the cell. So important role in evolution by facilitating recombination between axons of different genes. So this is the gene, we have axons, and then they will, by splicing, they will join together to form the protein A and protein B. So introns, the blue one will be removed out. Up to now, is it clear to all of you? Are you getting it up to now? Is it clear to all of you? So this is your homework also. You have to draw prokaryotic gene structure as well as eukaryotic gene structure. So please keep the homework somewhere so that you will be able to do it. Okay, just write it somewhere. So we have to remove the introns and only axons will be there, which is basically the main mRNA which will lead us to proteins like protein A and protein B. Now after it, again this is again the same thing, that gene is a sequence of a DNA which contains the genetic information which is required to make a specific protein. So Type of genes which we have prokaryotic genes as well as we have the eukaryotic genes. We have done it also. Now there are some important things also. The genes are, you know, they are just divided according to their activity. So we have different types of genes. The number one is housekeeping genes. The very number one is the housekeeping genes. So basically these genes, they always remain active. So these are those genes which always remain active. Then we have specific genes. Those genes which are getting active only during some special conditions, they are known as specific genes. After it we have basic genes. So these are the functional genes that bring about expression of a particular character. So basic genes that brings a expression of a particular character is the basic gene. Then we have lethal genes. These bring about the death, their processor. Basically lethal genes are, they just cause the death of that particular gene or particular cell basically. After it we have multiple genes. When two or more pairs of genes act together to produce a single phenotypic trait. Cumulative genes. Some genes have additive effects on the action of other genes. These are known as cumulative genes. Pleiotropic genes. The genes which produce changes in more than one character is the pleiotropic gene. So these genes which produce changes in more than one character. So you have to keep remembering this pleiotropic genes. Okay, because they change. These genes produce change in more than one character. Modifying genes. Inhibitory genes. The genes which suppress or inhibits the expression of another gene is known as inhibitory gene. So, as I already told you, it was Mendel in 1865 who assumed that there are factors. These factors are later known as genes. So promoting sequence is there just in front of the coding sequence, and at the end we have the terminator sequence which will stop the reaction. So this is minus 10 region, minus 35 region. At both these regions we have promoter sequences which will enhance the reaction or which will interact with RNA polymerase enzymes, which will increase the rate of transcription, or you can say for transcription or RNA synthesis we need these promoter sequences. So we have promoter, we have activator, we have enhancers. So this minus 10 is known as Krivinov box. Similarly in the eukaryotic genes we have exons, introns. We have its starting region, before it we have the promoter sequences. So these are the promoters. A promoter is regulatory region of a DNA located upstream controlling the gene expression. Okay, so we have cap which is 7-methylguanine at the 5' end, and at the 3' end we have the poly A tape. So you have to remember these things. Now some important features of a gene. Salient features of a gene. So number of a gene in each organism is more than number of chromosomes. Hence several genes are located on each chromosome. Because chromosome number is fixed. So this is chromosome. On these chromosomes many genes are present. The number of chromosome is fixed, but on a single chromosome or on each chromosome many genes are present. The genes are arranged in a single linear order like beads or a string. Each gene occupies a specific position and called locus. If the position of a gene changes, character also changes. So they have their own particular position. Genes can be transmitted from parent to the offspring. Genes may exist in several alternate forms called alleles. So genes are capable of combining together or can be replicated during a cell division. Genes may undergo for sudden changes in position and composition, which is known as mutation. Genes are capable of self-duplication, producing their own exact copies. So these are some specific features of a gene. that gene may undergo mutations also, they are able to replicate also, they have alternative forms which are known as alleles. Genes are transmitted from parent to the offspring. Genes are present on a specific position which is known as locus. So you have to remember all these points. So is it clear to all of you? The basic structure of gene is clear to all of you. Please let me know, because it is there in your syllabus. We have to cover fine structure of a gene, the basic structure of a gene. Similarly, I have one more PPT related to it also. This is again the gene structure. I will share it with all of you so that you will just go and watch it by your own self. Okay. So this is basically the gene structure, the fine structure of a gene. So is it clear to all of you? Please let me know. After it, we have to start another class or the session, the regulation of a gene expression. Regulation of a gene expression as well as lack of brain. So I will start with lack of brain and then I will let you about the regulation of gene expression also. So up to now it is clear to all of you. Please let me know. Everything is clear to all of you. We have done gene structure. So is it clear to all of you? Please let me know so that we will start the lack of brain. Please, any one of you. Yes, Devashish, how are you? So Devashish is my student from the previous batch. I hope he is fine. And they all are very good students of my previous batch. Yes, I am fine, Devashish. How are you all? Everyone is fine. Some of your classmates are in touch with you, like Juvelia, Judita, they are here. So how are you? Have your exams are completed or you have to, your final exams are still there? Hello. So yes, the structure, the fine structure of gene is clear to all of you. Please let me know so that I will start the next topic, which is basically the lack of brain. So we have to start lack of brain now, and the regulation of gene expression is also related with the lack of brain. So please let me know. Are you are you getting my points? The structure of gene is clear to all of you. Please reply. Yes, Devashi, you have your exam. Tomorrow, from tomorrow, okay. So all the best for your exams, Devashish. I hope you will do best in your exams. Definitely you will. And please be in touch. When you will be free, just be there along with me also for the sessions. Definitely you will get something new also. Okay. Okay, thank you Bhavni, thank you Sagarika, Ajay. So now we will starting with the next topic, which is lack of brain. Okay. So any one of you, have you done lack of brain before also? Please let me know, have you done lack of brain before also? Okay. Let me know if you have done lack of brain before also. Thank you Devashish, thank you so much for your support. And thank you so much. You are here. I feel so good today. Thank you so much. So now I am just going to start with the next lecture, which is on lac-oprene. After lac-oprene, we have to cover the regulation of gene expression. Now I am just going to start with lac-oprene now. Maybe many of you have done it before also, but I will try to keep it very simple, okay? So I will try to keep very simple the lac-oprene so that all of you will be able to get it. Yes. So, as we have just done the structure of a gene now, so you will be able to understand lac-oprene also. Let me tell you about the very basics which are there. So, first of all, the history of it, how the introduction, what is basically oprene, how it came into existence. So, oprene is a operating unit which can be defined as cluster of genes. Cluster means group of genes which are located together on a chromosome. So the group of genes, they are present together on a chromosome and they are transcribed together. Now what is transcription basically? When from the DNA we will get the mRNA. So the group of genes, they are present together and they are located on the chromosome and then they are transcribed together. So it is a group of closely linked structure genes and associated with the control genes which regulate the metabolic activity. So all the genes of the brain are coordinately controlled by a mechanism which was given by Jacob Monod and Lvov in Paris in 1961. You have to remember only one thing, that oprene is a operating unit which can be defined as a cluster of genes located together on a chromosome and they are transcribed together. So the group of genes which are present together on a chromosome, they will be transcribed together. So the lactosoprene, so the lac-oprene is basically lactosoprene. So what happens in lac-oprene? It codes for an enzyme which involved in the catabolism of lactose. So lactose, we have to show you the enzymatic reaction which is involved in the degradation of lactose. So lactose is the disaccharide. We all know that lactose is a disaccharide which is made up of glucose and galactose. It is the inducible oprene since the presence of lactose induced the oprene to switch on. So this is the basically the oprene is basically of two types, but it is the inducible oprene. So when the presence of lactose induces the operand to switch on. So when the lactose is present, this operand will be switched on. Let me show you. So this is again the gene, as we already discussed in the previous session right now. So gene, at that time we are just focused on a single gene. So from single gene we will get only the single product. But here the genes are joined together like Z gene, Y gene and A. They are joined together, and as always before it we have the promoter region. Before it we have some enhancers which will increase the rate of reaction. Getting my point? So we have promoters present, we have inhibitors also, we have Tata sequences also. So what will happen when the inducer is there? When the inducer is there, what will happen? Repressor inducer complex does not bind the DNA. So these lack this gene Z, it gives us beta galactosidase, Y gives permease and A gives transacetylase. So lacks that this gene codes for the enzyme beta galactosidase, and the function of what is the function of this beta galactosidase? It breaks down the lactose into glucose and galactose. So with the help of one enzyme beta galactosidase, it will be break down into lactose will be break down into glucose and galactose. Then we have lac Y. This lac Y, it gives the enzyme permease. So this protein found in the E. coli membrane actively transports lactose into the cells. Then the last is A, lac A, which promotes Trans-acetylase. Now what's the functioning of this trans-acetylase? The functioning of this enzyme is not known. So the functioning of this LAC is not known, but yes, the functioning of LAC Z and LAC Y is known. So I hope you will get it in this color. So we have operator which is before the sequence. Promoters are also before the sequence. We have repressor also. We have LAC-A1 also. So promoter will bind with the RNA polymerase enzyme and they will initiate the reaction. Please keep this thing in mind. Repressors are there. We also have repressors present. So repressor will repress the reaction. So repressor gene coding for LAC repressor protein binds to the DNA at the operator and blocks the binding. So what they do, it binds with the operator, or you can say with the it binds with the operator and it blocks the activity of promoter. So repressor plus operator, they bind together and they do not allow the reaction to proceed. So when this repressor, when this repressor binds with the inducer, they do not allow the reaction to proceed. So in the absence of lactose, or you can say inducer, when we don't have a lactose, the regulatory gene produces a repressor protein which binds to the operator site and prevents the transcription. If lactose is not there, lactose is an inducer. This is an inducible operating system. If lactose is not here, the repressor will bind with the operator site. It will bind with the operator site and does not allow the functioning of promoter. The functioning of promoter is basically to start the reaction, to proceed the transcription. So when repressor bind with the operator, it will not allow the reaction to proceed. So in the absence of lactose, when there is no inducer, the reaction will not start and the structural genes do not produce mRNA and proteins are not formed in the absence of lactose. Now when the lactose will be there, so this is in the absence of lactose. When the lactose is not here, the repressor will bind with the operator. So this is the promoter, this is the operator. So a repressor, this if you are able to see this yellow one, this repressor bind with the operator and do not allow the promoter to function. So if promoter will not do the working, how the reaction will continue? So repressor plus operator. Repressor binds with the operator and do not allow the promoter to attach with the RNA polymerase, and when RNA polymerase cannot bind with the promoter, how the reaction will proceed? So the reaction will be proceed in this direction. So when promoter will not bind with the RNA, how the reaction will proceed? So this is in the absence of lactose. Now when lactose is there, suppose the lactose is there, introduced in the medium, then lactose binds with the repressor. Lactose bind with the repressor. So when we have lactose, it binds with the repressor and it frees the operator. Now the operator is free. Getting my point? So the operator is now free and now the RNA polymerase binds with the promoter and there is the reaction which will continue. We will able to get mRNA and from the mRNA we will get the enzymes beta-galactosidase, transferases, transacetylase. So this mRNA codes for 3 enzymes necessary for the lactose catabolism. So this is the thing basically. So lactose is a inducer here. That is why it is known as inducible operand, because otherwise the reaction is not continue because the repressor binds with the operator. But now the repressor binds with the lactose, operator is free, polymerase will bind with the promoter and the reaction will continue. So we will get the mRNA and from the mRNA we will get the desirable enzyme. So enzymes needed to take up and use lactose are produced only when lactose is present. So this is basically the LAC operand system. So this is again the same thing, that when the repressor and operator will bind, the reaction will not proceed. The genes will not able to produce the enzymes. But when this lactose bind, what will happen then? We will able to generate the enzyme. So this is the RNA polymerase which will bind with the promoter region and the reaction will continue. So the reaction will continue. So in the absence of lactose, when lactose is absent, the repressor will bind with the operator and it blocks the RNA polymerase. So the reaction will not proceed further. But when lactose is present, it binds with this this repressor. Operator is free. Now, if you are able to see, this is lactose plus repressor. So now operator is free. RNA will bind with the promoter site and we will get the desired enzyme. So this is a thing which is there in case of operant system. So brain is a group of genes that are transcribed at the same time. So instead of one gene, three genes, LAC, Z, LAC, Y, LAC, A, three genes are transcribed at the same time. They control useful and important biochemical processes. One more thing, operands are there only in case of prokaryotes. So this is the operands basically which we have to study in case of prokaryotes, and the basic one is lac operand. So we have three genes which will code for three enzymes. Again the same thing, that when repressor is there, the reaction will not start. When the lactose will be there, the reaction will continue. So that is the whole thing in case of lactose. Then there are some mutations also, we will not discuss these mutations. But the main thing is that only. So I hope you will get it. Is it clear to all of you? The lactose is clear to all of you? Please let me know. The lactose is clear to all of you. Lacoprene. Lacoprene means lactosoprene. Is it clear to all of you? Any one of you? So this is your homework also. You have to draw the lacoprene model also. Like you have to draw the structures of the prokaryotic gene as well as a eukaryotic gene. Similarly, you have to draw the lacoprene system also in the absence of lactose as well as in the presence of lactose. Getting my point? So in the absence of lactose as well as in the presence of lactose. So this is your homework also. Is it clear to all of you? Please let me know. So any one of you please reply. Please, any one of you, you have to write this on Telegram group also so that all will able to answer. Is it clear? Up to now, is it clear to all of you? Okay. So shall we continue the next topic which is there, which is regulation of a gene expression. This is also related with this lack of brain. Shall we continue it? After it, your this 6, 7 and 8 topic will be completed. So shall we start the regulation of gene expression also? Please let me know. Okay, Shivani. I think that's all for too much. I think that's all for today. Then we have covered two topics. We have done fine structure of a gene plus lack of brain. So I will suggest to you. I will share the PPT also to go through it by your own self. Yes, yes. We can continue tomorrow. No worries. I just want all of you to be comfortable. The regulation of gene expression is also similar like this lack of brain. Basically, this is the lack of brain only. Just give me 5 minutes, Shivani, please. Just give me 5 minutes, then this will be over. Okay? Just give me 5 minutes more. Just give me 5 minutes more only. The topic is quite related with lack of brain. Only 5 minutes. Give me 5 minutes only. I am sorry for a little bit long, but the topic is quite interrelated, so just give me 5 minutes. It's only a small presentation. So this is the regulation of a gene expression, just we have done now also. So what do you mean by the term gene expression and its regulation? So what is gene, what is genome, total number of a gene expression, all these things are related together. So what is basically the regulation of a gene? The thing which we have covered in lack of brain, this is basically the regulation of a gene. Because we have some amount of lactose, we have some amount of inducible or suppressors which will enhance the reaction or stop the reaction. So this is the DNA. We have the promoter region, we have the Tata box which we have already discussed before. We have the promoter region on which RNA polymerase will bind and start the reaction. We have the protein coding regions which are known as exons, and at the end we have the termination. So this is in the nucleus. In the cell we have a nucleus. In the nucleus the double-stranded DNA will give us to mRNA. This is pre-mRNA. This pre-mRNA then gets changes into RNA, and this RNA then translated into the proteins. Now this is the main part which we have to cover. This is the main part which we have to cover. We have to discuss DNA replication, transcription and translation. Only these things are left. That is why I have covered this topic of the gene structure today, so that you will able to understand transcription, translation, replication in detail. So gene expression is a multiple process that result in the production of functional gene products. So we have different types of genes. We have housekeeping genes, we already discussed. Regulative genes are also there. Inducible genes are there. Gaseous inducers are there. These are only the types of genes only. Now, what do you mean by gene expression? How to regulate the gene expression? There are two types of regulations. We have a positive regulation, we have a negative regulation. What happens in case of positive regulation? Increased gene expression mediated by positive regulator. So, we have positive regulator which will enhance the reaction. We have negative regulators which will stop the reaction. So we have silencers or repressors which will repress or suppress the reaction. We have activators in case of positive regulation which will enhance the reaction. So these are the enhancers which are positive, and silencers which are negative which will stop the reaction. So this is the regulation of the gene expression. This is the DNA. From the DNA we will get pre-mRNA. From the pre-mRNA we will get the RNA, and at the end we will get the protein. We will discuss these points in detail. But yes, there is some negative control as well as positive control also. So gene expression can be modulated by control of transcription, post-transcriptional modifications, gene amplifications, gene rearrangements, control of translation, protein modification. So, gene regulation is influenced by hormones, heavy metals and chemicals. So, we have regulation in case of prokaryotes. So, we have operane there. Now, what are basically operanes? When we are combining 2-3 genes together, so genes involved in a metabolic pathway when present in a linear array. Cisterones are there. What are cisterones? Smallest unit of gene expression. which are known as cistrone. What are polycistronic mRNA? A single mRNA that encodes more than one separately translated protein. So three terms are there: operane, cistrone and polycistrone. Polycistrone is basically what? A single mRNA, single sequence that encodes more than one translated protein. So this is a brain model which is given by Jacob and Monod in 1961, also known as lacoprane model or lactosoprane. It consists of structural gene, inhibitor gene, promoter is also there, repressor is also there. So this is a diagram we have just discussed before, that if there is the absence of lactose, what will be there then there? The repressor will bind with the operator and the repressor will bind with the operator and promoter will not able to enhance the reaction. So lac Z give us beta galactosidase, lac Y galactosidase, lac A transacetylase. Promoter region is there, operator is there, inhibitor is there. So when inhibitor binds with the operator, the promoter will not be able to transcript the gene. So in the absence of lactose, we will not able to start the reaction. In the presence of lactose, we will able to increase enzymatic synthesis or we will able to start the reaction. So this is again the same diagram. Inhibitor or suppressor binds with the operator and the reaction will not continue. In the next step, what will happen? Lactose will bind with the inhibitor and reaction will continue and we will get the desired enzyme. Okay, the same thing is there. So that's only the thing which is there in the regulation of gene expression. This type of regulation is also there in the eukaryotic DNA, but that is little bit more complex. Our main focus is on the prokaryotes only or operand model only. So that is here only how we are able to enhance the reaction and how the reaction will be stopped. So I think that's all for today. Sorry for making it little bit longer, but the topic is quite related. That is why I think we will do it right now. Okay. So are you getting it or not? So Bhavna, please tag the syllabus. Please pin the syllabus so that all of you will able to get it. So I think that's all for today. You must get it definitely. If you will not be, then I will discuss it again also. No worries. But try to do it by yourself. If you will not get it, then I will help you. So that's all for today. Take care of yourself. Yeah, thank you. Thank all of you to be here for your patience. I think I have to thank all of you. Thank you all. So please take care of yourself. If you have any another thing to share, please let me know. So is it clear to all of you? Okay. Actually, you have to submit the review paper. You have to send me the abstract and the topic by 20th of December. I will let you know how many times you will get more. You have to submit it by 20th basically, but I will give you more time also. So if you have any another question, please let me know. Okay, thank you Bhavna, thank you all. Thank you for your patience and thank you for being here with me. And now we will meet tomorrow. I hope by this Sunday, I hope you all will be free from my side. We will cover the topics which are there. We have these two topics and then we have transcription translation. I will give more time to transcription translation, then we will continue it further. I will try to, but if not, otherwise by 15th of December, I will definitely try to complete it. Okay, by Monday or by Tuesday, I will try to complete your whole syllabus. So is it fine to all of you? Please let me know. Is it fine to all of you? That's all for today. Take care of your side. Till then, good night and please complete the assignment as soon as possible. All of you. So I think that's all for today. Yes, yes, America, today is the last date. But if you have some exams or something, you can take one more time also, okay? Yes, today is 10th. You are supposed to submit your review title along with the abstract by today only. So you have to submit it by today only. But still take your time and try to submit as soon as possible. Oh yes, yes, try to complete it as soon as possible, all of you. Okay, take care all of you, take care all of you. Submit your abstracts as soon as possible and please your assignment should be on time. I request all of you that your assignment should be on time. It's my request to all of you, please. So good night. Take care all of you. Please take care of yourself. We will meet tomorrow. In the meantime, try to complete your review paper as well as try to complete the assignment. Yes, yes, Ajay, there is no any worry. You can submit the abstract by tomorrow also. Please take your time, but try to submit it as soon as possible. I'm not forcing you to do it today only. You can take tomorrow also by tomorrow 11 a.m. or by 12. You can send me. That means in the morning you can send me. No worries. Okay. So good night to all of you. Please take care of yourself and we will meet tomorrow also. Uh, Actually, the assignments should be neat and clean, like you have already sent me the assignments of my topics and not which are very good. You are, presenting it very colorfully. I just watched it today also. So yes, your presentation should be good. That is the only thing. Like for today also, the review paper needs little bit more time. Just send me the abstract. Let me check it and then I will tell you. Okay. For assignments should be good. Assignment should be good. It should be well organized. Shwani, you can take more time for the review paper, but by up to 15th you have to submit me assignments continuously, and the assignment should be good because this is the only record which we have, your assignments only. So please try to do assignments on time and it should be good also. If you want to take more time for the review paper. No worries for the review paper, otherwise you have to submit the assignments on time. So that's all from my side today because I have my another lecture series. So take care of yourself. Good night from my side. We will meet tomorrow. Okay. So bye bye. Good night.